So we are on air, okay? I am recording. Okay, good. I hope it works. Doesn't tell me any. Do you see that I'm recording anywhere?
Yes, it's in the left upper corner.
Great. Thank you. Okay, so I will delete this part from the recording.
Can I ask something?
Yes, please. Go on.
Yeah, so I wasn't there yesterday because I had another lesson, but did you record the lesson yesterday also?
Okay, so I forgot to record the lesson. Okay. So blame on me for this. But if you go through the slides, the things that I talked about are very, very basic. Okay. So the issue about how the synaptic transmission work is not really necessary to understand the topics of this course, but it's good for you if you know a little bit how this works. Okay.
Yeah.
So please go a little bit through the slides and tell me if there is something that you really don't understand, and I'll be happy to explain that again. Okay.
I recorded the audio for, I think, at least the first half of the lesson. If you put your email in the chat, I can send that to you.
Yeah.
That's
okay. Thank you.
That's great. Thank you for that.
By the way, I wasn't there either yesterday, and I went through the slides, and the biggest thing I couldn't understand was the polarization. So if you could maybe talk about it like for 2 minutes, it would be great.
Sure. Sorry, I didn't understand the word.
The polarization.
The polarization, yes. So yeah, that's quite tricky. So just like this. Okay, so that's quite simple indeed. So the polarization. So neurons have a resting state. Okay. What does it mean? That the neuron where it is not firing, where it is not doing anything, like my slides right now. Our neuron. There you go. Okay, so the neuronal membrane. So cells have this membrane thing, right, which prevents any molecule to get into the cell. Okay, so you have these gates, which are basically big proteins, which are pumps. Okay, which filters the material that can get in and out of the cell. Okay, and you have several of these things. The most important for the synaptic transmission are these potassium, sodium, and calcium pump, because they regulate the interchange of electrically charged ions. Okay, why are these important? Because they can change the electric state of the cell. So as you see in the picture here, which my mouse pointer doesn't work. Okay, whatever. As you see in the picture here, you see that the inside of the cell is slightly negative and the outside is positive. Okay, this is called the resting state of the cell, and it's about minus 70 millivolts. Okay, so it's negative. Then what happens at some point is that you have a depolarization. Why is it depolarization? Because by standard, something polarized is something negative. Okay, so if you depolarize it, you turn it more positive. And then when that goes from minus 70 to minus 50, then you have something that changes abruptly. Okay, so what happens after the sodium channel open is that you have this little polarization from 70 to 50, and then the membrane becomes completely permeable. Okay, and then you have this sudden depolarization that goes from minus 50 to above 0. Okay, that is the spike. That is the action potential. When the voltage goes beyond 0 and the charge inside the cell is reversed. So now the inside of the cell is positive, right? And this happens in every single cell which is present, in every single cell, in every single piece of cell which is basically on the axon, right? So you have like a sort of a domino effect of all these ion pumps opening and closing, and they propagate the electric signal, okay? Which is positive, which is above 0. Then, when the spike is done, the potassium pumps start working, and they restore the negative voltage inside the cell by kicking out potassium ions, which are positive, right? So eliminating the positive charges, the cell becomes negative again. So it goes from negative to positive to negative, okay? Now, a nice example is that some modulation, for instance, some other transmission, some other neurotransmitters could induce the cell to be more excitable or less excitable, okay? More excitable means that the resting state goes from -70 to, let's say, -60. So then it's easier to bring the threshold, to bring the level up to the threshold, which is -50, right? Or some neurons would transmit inhibiting modulation to the cell, and then it becomes, let's say, more negative, let's say -90. And then you need more positive electricity, more signal in order to trigger the spike. Okay? Was this clear? Is there any other
Yeah,
questions?
it's clearer. It's clearer. I think I understood better. Thank you.
Okay. So the cell is mildly negative, and then when you have the spike, it switches from negative to positive, and then it goes back to negative. And this is just one spike. You can have like 40 of these spikes in a second, for instance, or you can have 10, or you can have 20, right? And they are measured by hertz, which is vibration per second, or spike per second, okay? So if you have firing at 40 hertz, it means that you have this spike going on in a second 40 times, okay? Good. So this is basically the action potential, which is the output of the neuron, right? But then this output gets to the synapses, and then from the presynaptic terminal, this output, this electricity, triggers the release of neurotransmitters, which are chemicals, and you can see them here in the picture. And then they bind to the receptors in the receiving cell, in the soma or in the dendrite, okay? And these receptors trigger the same electric reaction, a similar electric reaction in the receiving cell. They do not trigger an action potential. They trigger a gradual depolarization. And this gradual depolarization is strong enough. The receiving cell, in turn, will trigger an action potential, okay? An example of this is this. When I want to move my finger, I'm sending a signal from my motor cortex to the interneurons in my spinal cord, and those interneurons are going to take the signal.
Also wie ich reingekommen bin, war alles zugesperrt.
Sorry?
Schau mal, ja.
Oh, okay, you're not talking to us. Are you? No. Okay, good. So it in the motor neurons, it gets the spike from the let's say the primary motor cortex, and then it activates and it retransmits another spike to the muscle, right? So in two synapses, you go from your brain to your muscle. Okay, so this is an example. Okay, so if this is clear, let's continue, okay? And let's continue with this. If you remember, we talked about the fact that the brain, different parts of the brain seems to be specialized for different functions, right? And how do we know this? So now we're gonna talk a little bit about this, but before knowing this guy here, Brodmann, in 1909, made a very, very important discovery. He created a cytoarchitectural map of the cortex, okay? cyto means tissue, basically. So a tissue is a layer, is a set of layers of different cells, okay? Like the cortex. The cortex is a tissue. The skin is a tissue, okay? It's not just made by one layer of cells, it's several layers. And he basically noticed that our brain is like a map of different patches of tissues that look different on the microscope. Okay, and you have the original drawing of Brodmann right here. So he basically divided the whole cortex in different areas and he numbered them. And we are still using this numbering because this was really, really, really smart. Okay, so each part of our brain is actually morphologically, anatomically different. And these little patches, they are kind of more homogeneous inside, within themselves, and they're more different from other parts of the brain. These are called brain areas, BA, okay? BA stands for Brodmann area. Right, so the question is, why do brain areas have these different cytoarchitectonic properties? Right? So again, if you look at the brain, this is the different brain areas in Brodmann subdivision, okay? For instance, Broca's area is BA44 and BA45, and some people also add BA47. Wernicke's area is two different areas. So it's a little bit of the STG, which is, I think, BA30 something. I don't know exactly which one. Should be 39, probably. No, 22, sorry. And then a little bit of the supramarginal gyrus, which is BA40, I think. But I'll tell you more precisely. Now it's not important. So I haven't told you why brain areas have different architectonic properties, but let's see why they have it, right? So something that I haven't told you is that a cortex is usually divided in 4 layers or 6 layers if it's a neocortex. If it's that part of the cortex that only we and very advanced animals like primates have, okay? So if you take a cortex, a neocortex with 6 layers, you see in the picture that every single layer has different population of neurons, which have different morphology. So if you see at the level 5, which is very important for the input and the output of the cortex, you have these pyramidal cells, which have a long dendrite and then a long axon, okay? But then if you look at the second layer, you have much, much smaller cells called granule cells. If you look at from the 5th to the 6th, you have cells with different shapes. So this is kind of important for processing information, for instance. And then the 6th layer merges into the white matter. In some areas, it merges abruptly. So you see the difference. In some areas, it's more smooth, the transition, okay? Now, again, you don't have to know the details about this, just to tell you that the cortex is composed by 6 different layers of cells, but we still didn't know why, right? So in order to understand why, we'll have to wait 40 years and to dive into the pioneer discoveries by Penfield. So who's Penfield? Penfield was a guy, was a neurologist, basically, who decided to electrically stimulate the cortex in vivo while people were awake, especially while they were undergoing brain surgery, usually for epilepsy, okay? So what did he do? He just stimulated with very low voltage electric current, the cortex. Now you might think, oh, this is really, really, really painful. In fact, it is not, because the cortex has no pain receptors. So you can do whatever you want. So he stimulated 400 living and awake patients during surgery. And then he was, asking them what they would see. And some of them said, when he stimulated the occipital lobe, they would say, oh, a star came down toward my nose. if you stimulated the central sulcus, they would say, those fingers and my thumb gave a jump. If you stimulated the temporal lobe, they would say, I hear music again, someone who was deaf. So what does this suggest us? That if you stimulate the different brain areas, you sort of have a feeling or a perception. What do you think? Why, if you put electricity in your occipital lobe, you would say that you see some light or some star?
Yeah, I mean, it kind of demonstrates the specialization of the particular part of the brain that you're stimulating. So the occipital lobe, I think, was the visual.
Right, exactly.
I remember.
Exactly that. So I found the most convincing evidence that our brain does not work as a unit, as a holistic thing for specific processing or processing specific information. But again, functional specialization is the rule, okay? So by doing this, for instance, if you zoom in your somatosensory cortex, you're gonna see this. You're gonna see what's called a somatotopic map, where that it goes from your genitals throughout all your limbs to the face up to the guts, the intestine, and these kind of things. You see that? That's called homunculus. It was a major discovery to see that the parts of our body, of our sensomotor cortex, the one that computes the information when you touch your body, that it has this map, which is directly mapped on the cortex. So by looking at these homunculus, what can you tell? What can you tell about, for instance, the size of different parts of the body?
The size is probably correlated with the number of receptors.
Exactly. So you see, for instance, your face is disproportionate compared to the other parts of the body because your face is very sensitive, because you have a lot of sensorial receptors on your face. For instance, the lips are very important for instance, sensing the kind of food that you're eating, right? There is a very important evolutionary function. Therefore, your lips on your cortex take as much space as let's say, part of the leg, or almost the foot, or let's say, I don't know, the forearm. Okay. So that's because lips are very important. Okay. And you see also the thumb has way more receptors than all the other fingers, and the fingers at the arm has way more receptor than the hand, right? And so on and so forth. And you can, so if you have a nice representation of the homunculus, that's the guy. Huge hands, huge lips, huge face, small body. Okay. Right. So if we have a general picture of the cortex, that's basically how it looks like. So you see now, if I do this, do you see my mouse? Yeah, good. So you see now that the areas we were talking about, for instance, the Broca's area right here in your frontal lobe, is very, very close. In fact, it almost overlaps with the primary motor cortex, which makes sense if we think that if you have Broca's area damage, you have problems of producing language. Well, then when we go to the Wernicke's area, it is in fact included in the auditory association cortex, okay? Which is very close to this little blue thing, which is your primary auditory cortex, okay? So you have a gradient of information processing from primary area, which process the primary information of your sensorial input, to associative or secondary areas, which makes higher order computations, okay? For instance, the primary auditory cortex might process the intensity of the sound, the pitch of the sound, but then the phonological reconstructions, what tells your brain that B is different from T, happens in the associative area, okay? This is not entirely true, but it's like an example. And so many people claim the Broca's area is in fact a supplementary motor area, a secondary motor area, but this is, we'll see, this is not entirely true. All these high-level areas, they're better thought of as association cortex, okay? So anything that we don't really know what it's doing is basically dubbed higher level multimodal association cortex, okay? And you see the same thing with vision. You have a primary visual cortex, and then you have a lot of secondary cortex. Some of them are processing movement, some of them are processing color, some of them are processing shapes, some of them are processing faces, okay? And each single part of your brain is specialized for one function. Good. So then again, a little look again at our most important areas. The areas that we are gonna talk a lot about is this, the inferior frontal gyrus, where your Broca's area is, the superior temporal gyrus, where you have your lexical and phonological processing, the supramarginal gyrus, phonological processing, part of Wernicke. The angular gyrus is really important to integrate any kind of information coming from your brain, especially visual information with temporal information, with conceptual information. And then we are going to talk about a little bit about the anterior temporal lobe, which is not written here, but the temporal poles and all the three layers of the temporal gyrus, especially the middle temporal gyrus is very important for semantic processing, okay? Good. So this is it for our introduction. Any question about the structure of the brain or neurons or stuff like this, or can we go on to the second part? I guess we can proceed. Let me see if my, oh, this is still stuck. Okay, whatever. I'm gonna use, I'm gonna use Keynote, whatever. All right, so now we're gonna talk a little bit about methods in neuroscience. So most of the stuff we're gonna talk about are experiments or studies conducted with 2 or 3 methodologies, okay? One is fMRI, one is EEG, and the other one is MEG. Now I'll tell you what these things are, okay? Is any of you very familiar with these things? Is any of you very, very experienced in these kind of things? No. Good. That's good. I'm not gonna speak for that. Now, let me show the chat and show manage participants. Okay, good. All right, so so the first thing that I want to show you is this picture, right? And this is a nice picture that tells you a little bit why do we have different methodologies? Why cannot we use just a single methodologies? Well, the idea is that each one of these methodologies have some pros and some cons, okay? Especially due to their very functioning. So for instance, if you look at fMRI, functional MRI is this orange square here. It has, this is a spatial resolution that goes from 0 to 3 millimeters, okay? From the level of a neuronal column to a level of the brain, which is not super precise. If you go to, for instance, single unit recording, then you can record one single neuron, and that's more precise, okay? As for the temporal resolution, which goes from milliseconds to days, okay? If you look at lesions, natural occurring lesions, then you're gonna see a resolution of days. But if you look at functional MRI, that goes from 0 to 3 seconds, roughly, okay? So within the order of seconds, which is kind of fine if you think about what happens in the brain when we read a sentence, because a sentence would take like 3-4 seconds to be read, right? But if you want to know exactly what happens as soon as you hear a word or a sound, 3 seconds is too much. You're not going to have enough resolution to see exactly what happens. You need something faster, something quicker, and that's where ERP and MEG come, right? You see that this square, this purple square, is actually on the left side of the fMRI because they're faster. Unfortunately, the space resolution of ERP is lower than functional fMRI. It can get kind of to the same level, but it's much less rich. As we see, with fMRI, we'll see exactly which part of the brain is active when we do something. With EEG, you only see that if you do a lot of additional processing, and it's not that reliable. So you see the trade-off. With EEG or ERP, ERP is a particular technology of EEG, right? You have a higher time resolution, a higher temporal resolution, but a lower spatial resolution. So if you're interested in time, then you should use that. If you're interested in localizing stuff, then you should use something else, okay? Everything clear?
Clear.
Okay. Is there a question? No. Good. So before diving into the technologies, I just want to tell you a little bit about some basic concepts about representations, okay? So what are mental representations? I'm going to give you some examples. Sounds are mental representations, okay? Written words are mental representations. Objects are mental representations. Can you tell me more? What could be another example of a mental representation?
A concept.
A concept, very good. Other stuff? For instance, yes?
A smell?
A smell, yes. So all the sensorial representations are mental representations, right? And you might say, this is not true. When I smell stuff, it's because there is smell around. Well, you know, when you smell stuff, it's because there are little molecules of stuff that are getting to your receptors in your nose. And those receptors create the feeling of smell. There is no smell in the real world. There is no colors in the real world. And in a way, there are no sounds and there are no objects even. Now, you might think that I'm saying crazy things, but we are going to talk about this in detail, okay? Phonemes are mental representations. You have something like sounds, but you don't have phonemes in the real world. Memories, right? There are no memories in the world. Phrases, beliefs, okay? All this stuff is our mental representation, which means that they are made up by our brain. Thoughts. And then you can see there is a gradient from less abstract to more abstract, because a sound is relatively less abstract because they have a very, very connection to its physical properties, which we're going to talk about next class. But a thought is a collection of images, sounds, memories, feelings, you know, what else? All right. So one of the main aims of neuroscience is to map mental representations onto neural representations, okay? What are neural representations? They come from the functioning of neurons together. And we know that these neurons are actually creating those representations, because if you have those neurons damaged, you don't have that representation anymore. You cannot access it, okay? So you have many different kinds of neural representations, right? You see all these nice diagrams, but in particular, we can summarize the neural representations in 3 kinds. One is so-called local representations, which is basically one neuron, okay? Distributed representations. A single representation is distributed in a network of neurons, in a huge network of neurons, thousands and millions of neurons. And then, yeah, so all the information about the stimulus is carried in the whole population. A single neuron is not going to encode any specific representation, okay? But just a subset of the information of that representation. And then we have an intermediate layer, an intermediate concept, which is sparse distributed representations. A small proportion of neurons carry the information, okay? You see this graduality of things. Now I'm going to give you an example, which I particularly like. So you might wonder, yeah, but like, why are you saying that one neuron can carry the whole information about one representation? What is the evidence for this? Meet the Halle-Berry neuron or Hale-Berry neuron in the medial temporal lobe. So this guy, Quiroga et al. In 2007, he found one single neuron in one person that only responded for you say Hale-Berry or Halle-Berry? I think Hale-Berry.
Halle.
Sorry?
Halle-Berry.
Halle-Berry, thank you. Who you might remember from Catwoman in one of the last famous Batmans, okay? Now, the interesting thing about this is that this guy found that this neuron would fire, and now you know what that means, right? Would fire action potentials every time the subject would see Halle-Berry dressed up as a Catwoman, but not other Catwomen, the name of Halle-Berry, but not other names, different angles of the picture of Halle-Berry, but not other pictures. So, can we conclude from this that this neuron is only sensitive to Halle-Berry? What do you think?
Oh, yeah.
Yeah, someone might say, yeah. This guy, for instance, said, yeah. But like some guy who's more picky might say no. Because in order to derive this conclusion, we need to test millions of stimuli, which all represents Halle-Berry, and millions of stimuli which does not represent. Maybe this neuron was just coding some specific information about actresses that this guy liked that saw that particular day, right? But there is something to it, especially the multimodality of this neuron that can answer for a written name, can respond to a written name or to a face or certain things. Okay, this was just an example of a local neuronal representation. Good. All right. So now that you know how neuronal representations can be thought of, let's move to EEG, one of the most important neuroscience technology. Used a lot in the last 50 years, and I'll tell you why in a second. How does an EEG work? It's very simple, okay? You have these guys here called electrodes, which are placed on your scalp. What do these guys do? They record electric current. Why is there electric current on your scalp? Well, we just seen that neural transmission is basically electric transmission, is electric current flowing in your neurons. At some point, it turns into chemical energy or chemical information, and then it's turned again in electric current. So there is a lot of electricity, very, very low voltage electricity going on in your brain, and this electricity, in a way, it's measurable from the scalp. Okay, so that's the electroencephalogram. And how does it work? It measures the electrical currents generated by the brain. Now, if you know a little bit about electricity, and I didn't, you might know that you cannot measure any electric field or any electric current if you just put one single electrode. In order to measure a current, you need 2 electrodes, right? Also called dipoles. So what you measure is the potential difference between two different electrodes. Okay, think about, I don't know if you ever seen the little thing to measure the electricity in the batteries. You always need two cables, right? Because you need to close the circuit in order to measure the flow of electricity. So that's what the EEG does, and what you get is these little waves. So for every electrode, you get different waves. So how do you know, for instance, that this electricity is different from this one? You have one electrode here, but where is the second electrode? The second electrode is called reference site, okay? It is usually put in a neutral position, your mastoid bone, right here, because you're not supposed to have cognitive processing going on in your bones. No one knows for sure. Another possibility is to use the average reference as a reference. The average reference is making an average of all the electrodes and using that as a reference to one single electrode, okay? Different people use different standards, but these are the two most important ones. And it records the electric voltage at the scalp. Now the question is, what does this electric voltage reflect? Is it the working on one neuron, two neurons? Is it presynaptic potential or postsynaptic potential? Is it everything? Different parts of the brain, only the cortex, the action potential, or the dendrites potential? It records the postsynaptic potential rather than the axonal current, okay? Why? Because we know that the action potential are very short-lived. They're like an instant burst. On the other hand, and they rarely fire together. On the other hand, the postsynaptic potential lasts from tens to hundreds of milliseconds. You remember the gradual spread of electricity in the dendrite throughout the whole receiving neuron. And also, given the parallel organization of pyramidal cells that I told you about, right? Their activity sums up for spatial properties. So, in order for any brain electricity to show up in the EEG, what do you need? Several thousands of spatially aligned neurons firing, receiving, in fact, signal at the same time, okay? It's what is called a synchronous activity of a large population. You're not gonna find anything, it's just a few neurons fire, okay? There you go. Here's your electrode, right? Which is just a sensor, nothing more than a sensor. And that transmits the signal to an amplifier, which amplifies the signal. But then look at how much crap you have between the neuron and your electrode, okay? In particular, you have 3 layers of meninges and a 4 layer of cells, and your skull, unless you broke your bone, and the skin, your skull, okay? So all these guys are gonna interfere with your electric signal. In particular, they're gonna be absorb or distort your electric signal. This is why if your electrode here captures some current, doesn't mean that the current is generated by the neurons right above, but it could be far away, okay? So here are two problems of EEG. The voltage must be strong enough, and the skull and the meninges attenuate the signal, okay? So you have a lot of distortions, right? And also, there is another problem, which is about the shape of your cortex. You remember the cortex is made of bumps called gyri and grooves called sulci, okay? The problem is that these two things are gonna cancel out their electricity, because one is transmitting a flux of electricity in this direction, the other one in the other direction, and they cancel out each other. So what you're gonna get is only this, okay? The dipoles sum together with aligned neurons. Those who are not aligned either are gonna show up somewhere else or they're gonna cancel each other, okay? Then important, electrodes are placed at standard positions. So then you can compare different experiments on different people, right? So the central line is called Z, and then you have even numbers on your right and odd numbers on your left. Okay, phi P is frontoparietal, F frontal, C central, P posterior, O occipital. All right, just like everything. Good. Now, the next thing I'm going to tell you is that if you look at the track of the EEG signal, and we're going to look at a few of them, this is going to be a little bit of, let's say, an envelope, okay? So with some highs and some lows, like a fluctuation of current. Now, the first thing you're going to see is not actually what you're interested in. They are evoked potentials. The evoked potentials, they follow any external stimulation, like visual stimulation or auditory stimulation, and they are stimulus dependent. Not stimulus dependent, but modality dependent. So for instance, I'm going to show some examples. But what we are interested in is the event-related potentials, which is associated with a single event that is exactly what you're going to present to your subjects, which could be, for instance, a word, a sound, picture, spoken word, or a written word, right? And then you're going to measure exactly what happens right after you present your stimulus. That's why they're called event-related or event, because they are time-locked to an event. And they're triggered by, I mean, this is kind of weird, but stimulus-independent cognitive processes, meaning that, for instance, they could be triggered by lexical access, and you have lexical access for a lot of different stimuli, okay? Attention, memory retrieval, language-related processes. So now you might be wondering, what are the first kind of evoked potentials? These are them. So you see, for instance, this is a picture of recording in a mouse, and this is a human, okay? And this is the recording of the evoked potentials after the presentation of a sound. And what you're going to see is this: up and down and up and down and up and down and up and down and nothing, okay? What are these up and downs? Well, these represent the different stages of processing of the auditory signal from your ears or from your cochlear nuclei to your brain cortex, right? And you see, comparing the mouse and the human, the timing is very, 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 very similar. So these little things, they are just a reflect of the automatic working of the cortex, especially which goes from your ear to your thalamus, to your nerves, your ear, to this nuclei in the cochlear, which is your part of your ear, to other nerves, to the superior olivary complex, which is a kind of sound processing thing, to your inferior colliculus, finally to the thalamus, which is the medial geniculate nucleus of your thalamus, and just way after that to your cortex. So in order to get the signal to your cortex, you will have to wait a few tenths of milliseconds, okay? Now, I don't have the time scale here, but it should be, I would say, something between 50, 60, something like this. Yeah, it should be written somewhere. Okay, now, this is how it looks like, an EEG signal in some electrodes, okay? And you see all these, again, waves up and down. Now, neuroscientists wanted to make your life harder, and they decided to switch the voltage. So what is up is negative, and what is down is positive, okay? Some people use the opposite convention. We're going to stick to this one because that's the one that you're going to find in papers, okay? So you see this P1, it's a positive deflection of your signal at about 100 milliseconds. Is it due to the word that you're reading or hearing? No. It's a general spatial attention signal generated in your extra-striated areas, your secondary vision area, at about 100 milliseconds. Before that, if you have a visually presented stimulus, you're going to find something similar called C1, cortical visual component, okay? And then you have the N1, which is a different voltage, which comes from temporal and parietal generators, and that is sensitive to attention. The more attention you're paying to certain stimulus, the higher this signal will be generated, okay? Is there any question about this? This is not really important for our purposes, but it's important to see why you have these up and downs when you look at the signal. They're just not random. They're different areas of the brains generating different electric currents, okay? Now, let's look at what is interesting for us, which is the event-related potential. So I told you that the ERPs, in the ERPs, there is a lot of noise, okay? So if you look at a single track, you're gonna get some messy stuff like this, right? But then if you look at the average of 10 different tracks, let's say 10 different sentences, all average, then you start seeing the evoked potentials. If you have 50, it's nicer. If you have 100, it almost looks like a beautiful ERP track that you would put on a paper, okay? That's why you need many trials, many sentences, many words for every condition, for every experiment, okay? Between 40 and 100. In order, the more trials you have, the more averaging of trials you're gonna do, the nicer and smoother and less noisy your signal is gonna be, okay? Now, there are some statistical techniques that allow you to work directly on the single track, but we're not gonna talk about that. So we assume that when we're gonna look at ERPs, this is gonna be the result of the average of many, many, many different trials, all time locked at the same point, which is usually the onset of the critical word, okay? Here's one example, most famous example for semantics. So people were hearing these words, or in fact, reading them one by one on the screen, okay? It was his first day at work. Good. Compare this to he spread the warm bread with socks. Spreading warm bread with socks? Surprise. Weird stuff. The semantic anomaly or semantic unpredictable surprising continuation due to socks, because you would think that you would spread the warm bread with a knife, right? No, sorry. Butter, or a knife. I don't know. Any English speaker here? I don't know if it's the instrument or the substance you spread the bread with, whatever. Right. So now let's ignore the third condition. It's not really interesting for us, right? What she found is that when you have, so they had a lot of different sentences all matched by the length of the word and other properties of the word. What she found is that when you have semantic unlikely continuation or semantic surprise or semantic anomalies, you get this negative deflection at about 400 milliseconds from the onset of the word, which is called N400. N because it is negative, 400 because it happens at around 400 milliseconds after the onset of the critical word, okay? And this is a component. You see it it was butter, yes. Sorry about that. So here is another study. You see that when you get butter, you have a negative deflection compared to the baseline, which is, sorry, the other way around. When you have socks, you have a negative deflection from the baseline, which is butter, okay? This big wave. This is the N400. And it's interesting because it is a component. And being a neuropsychological component, it has some properties that are quite stable across experiments, across different words, and across different sentences. And these properties are the latency and the distribution. Okay, distributions means where it is on the scalp, which electrodes are gonna record it. Latency means the onset. Sorry, the time that it takes for the signal to show up after the onset of the critical stimulus, of the critical event. Okay. So, and these components, since they are so stable, they are associated with cognitive processes. So if I told you that the N400 is coming up when you perceive an unpredictable, surprising, weird word or weird sentence continuation, what is the process, the cognitive process that is associated to this component, that modulates this component? What do you think it is? Is it phonology? Is it a visual process? Yes?
The semantics.
Something about semantics, right? It's probably something about semantics. Yes, it is the most important semantic, like ERP component. ERP, event-related potential component, these guys, okay? If you want to talk about syntax, then you probably want to talk about the P600. Now you see that the P600 is different from the N400 for two main properties. First, it is P and it's not N. What does it mean?
Positive and negative.
Yes, very good. So it's a positive deflection. And it's 600 and not 400 because it has its peak at 600 milliseconds. Does it mean that syntax is lower than semantics? Some people would say yes, but absolutely no, right? This is just one component that you see, and then you can see many, many other components, and we are going to talk about that. Problems. What is the problem about all this? So let's say that you have two, I mean, what you get at the end of the day is just a signal in one electrode, right? So what if you have two components interacting to each other? What are they going to do? You don't know. You only get this at the end of the day as a result, and this signal, this EEG signal, or this ERP average signal, could be due to these three components reflecting three different processes occurring at the same time. See? One causing the first bump, one causing the second negative bump, and one causing a third positive bump. But it could also be this one. It could be that this component is much, 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 much bigger than the C1, but the C2 is also much, much, much more positive, and they cancel each other. So how do we know? It's a very, very tough question. The simple answer is that we never know. But then, right, so keep faith and replicate, or use some very, very fancy source localization or source reconstruction methodologies that cross the information from all the different electrodes, and they tell you basically what is the most optimal solution to this problem. Okay? So again, pros of ERP: high temporal resolution, it's super fast, order of milliseconds. It's cheap. It's only 25,000 for a system. You might say, well, that's a lot of money. Okay, but an fMRI is like millions, right? So it is cheap. There's a huge experimental literature, more than half a century. Cons: poor spatial resolution, no comparison with fMRI. The inverse problem: you get these signals coming from all the electrodes, but you don't know what's the generator of these signals. That is called the inverse problem. It's basically the solution of an equation with many different equations, each equation for each single electrode, right? One equation for each single electrode. and there is no single solution. Right, so we know the electrical potential of the scalp, and we must infer the number, the location, the magnitude of the electrical sources, all right? But there are interesting toolkits to do this. And this is called ERP source localization, for instance. This is an example, and you see the localization was right here in your temporal cortex, okay? Now let's keep this one and let's go to the MEG. I'm gonna say a few things and then I'm gonna give you 5 minutes of break, okay? The MEG is very, very similar to the EEG. The difference between MEG and EEG is that while EEG records electricity, the MEG records magnetic fields, because every current, like this one, generates an orthogonal magnetic field. So your current goes on the two-dimensional space, your magnetic fields pops up like this, or better, like this. It's something that turns. You see that right here, right? The yellow thing is your current, the green thing is your magnetic field. So why bother? Why do we have to buy a much more expensive machine? Well, because the MEG has some cool stuff. For instance, a much better spatial resolution. It is expensive, but it is more sensitive at the sulci, whereas the ERP is sensitive to both the sulci and the gyri. And it is poor at detecting deep dipoles, whereas the EEG is better at detecting deep dipoles. Dipoles are generators. Deep means in your thalamus, for instance, or in the deeper part, in the ventral parts of the brain, okay? And the cool thing about the MEG is that unlike the electric signal, the magnetic signal, because of its physical properties, which I guess is about the length of the wavelength, it is unaffected by the skull and by the meninges. So it is more, in a way, more reliable as to where the source of the signal comes from, okay? So you see, pros and cons. Why MEG is cool? Very high temporal resolution, very high precise localization, okay? For instance, this guy, Pulvermüller, presented sentences involving arms and sentences involving leg. And then he found this kind of somatotopic or semanticotopic different activations. These were the arm related and these were the leg related on the somatosensory cortex. So he found that when you say kicking a man, you're going to activate your somatosensory cortex of the leg. When you say kicking a ball, slapping a man, then you're going to activate the area of your hand, okay? And this was seen with the MEG. Forget about getting anything like this with the EEG. Okay, so what you're going to get with the MEG is like this. It is similar to the EEG, right? You get this kind of signal, and then you have to reconstruct the dipole, and then you can have one dipole, two dipoles, so it also depends on your assumptions, okay? But with sophisticated programs, let's say, you can do that. Good. So 5 minutes break, and then we do fMRI, okay? Okay. And we can start again. Are you all back?
Yes.
1, 2, 3, 4, 5, 6, 7. All right, good. So the fMRI is one of the most important methodologies because it has been used over and over and over because of its very, very good spatial resolution, okay? And also it's good to see what happens in people that have a lesion, for instance, which areas are active in people that have a lesion. But the fMRI wouldn't be possible if the MRI wasn't invented, and that's what we're going to talk about now. So the MRI is like a very, very powerful TAC or TC, right? So, you know, when you do an X-ray, you get a bidimensional picture of your bone. If you do a three-dimensional X-ray called CT, sorry, computerized tomography, okay, then you're gonna get a 3D X-ray picture. But this is even more sophisticated. Why? Because it's way more, you have a way higher resolution of a picture. That is called MRI. Some of you might have done an MRI to a knee, for instance, if you broke your knee. Did any one of you ever done an MRI?
Yes.
There you go. Can I ask you what?
Head.
There you go. So you saw the picture, like the very, very detailed picture of your brain, right?
Yes.
Right. So you know what I'm talking about. For those who's never done it, I'm gonna show you a lot of nice pictures. That is called structural MRI. So what you see in the MRI is the brain, the structure of the brain, the white matter and the gray matter. How does it work? It does not use radiations, so it's safer, very high resolution, and it sees gray and white matter and brain structures. And that's what you get. With different trimming and different methodologies, you get different pictures, right? So in the picture in the middle, you see nicely the white and the black matter. In the picture of the right, you see the water, which is inside your ventricles very well, right? So these are different weighing of the fMRI. How it works? It works with the protons that are in the water molecules in your body. Protons have weak magnetic fields, right? The MRI scan is nothing like a huge, strong magnet above your head, which ranges from 1.5 to 5 Tesla. Tesla is a unit of the magnetic power. To give you an example, the Earth magnetic field is 0.0001 Tesla. So these are super strong magnets, electrically induced magnets, okay? What happens is that when you put this strong magnetic field on your protons, on the protons of the water, of the molecules of your brain, they're gonna do like this. They're gonna, small fractions of protons will align to the magnetic field, like this, all right? And then you apply a brief radio frequency, a pulse, which will knock the orientation from vertical to the direction of the pulse, which is horizontal in this way, right? So some of them are vertical because of the magnet. You send the pulse, they're knocked by 90 degrees. What happens then is that after some time, when the pulse is done, they will relaxate and they will come back to their original position. And this process, repeated many times, is going to give you a slice of the scan. Why? Because when they turn back to their positions, right, they are gonna perturbate the signal, that the magnetic signal that you are recording. So if you apply different technique, like for instance T1 or T2 or T2 star, you are basically applying different time frequencies to the signal, and then you're gonna see a different output, okay? The variation in the rate at which protons can return back, they are used to distinguish between different types of tissue, basically. Okay? So this is, you know, just how it works. It basically works on measuring what water molecule does due to their magnetic properties. Now, this is interesting, but it only tells you information about the structure of the brain, right? What we want to see is the function of the brain. What's going on during a stimulation. So from the structure, we have to move to the function. And how do we do that? Well, we had no idea how to do it until this very smart guy, Sergi Ogawa, invented a technique called BOLD, blood oxygen level dependent. And this is mostly used in the high majority of fMRI studies. This exploits the properties of hemoglobin. Do you know what hemoglobin is? Can someone tell me?
It's the particle that attaches the oxygen. It's the red coloring.
Exactly. And it's a particle of your red blood cells, right? They have this important molecule that has this property of attracting oxygen or CO2, right? So when it has CO2, we call it deoxyhemoglobin. This guy found that some perturbation in the signal of the normal MRI were due. Some people thought it was just noise, but he said, well, maybe this is not really noise. Maybe this is induced by something. And he found out that this was induced by deoxyhemoglobin. So the blood cells bring in CO2 to your lungs in order to be expelled by your breathing. All right? So the oxyhemoglobin is diamagnetic, it has no distortion. The deoxyhemoglobin is paramagnetic. It induced distortion of the magnetic field. In particular, it induced darkening of the signal at T2 star, okay? Now, by exploiting these properties, we can measure how much brain is active depending on how much oxygen it is burning or it is demanding because of a cognitive effort, okay? But how? That's what I'm going to tell you now. This was one of my favorite cartoons. So the red blood cells take oxygen from the lungs, oxyhemoglobin. Our brain uses 20% of our oxygen budget. I told you 40 last time. It was too much, okay? It's actually 20. So it's a lot actually, because the brain doesn't really move, doesn't have muscular contraction. So the red blood cells, after the brain used that oxygen, okay, they're loaded with CO2 and they're bringing it back to the brain. See this, to the lungs, sorry. These blue bubbles here in the cartoon. Some of them have some oxygen, some of them has some CO2, okay? Before going on, can you tell me why and where our brain is using oxygen? That's a very tricky question, but please, yes.
It's burning it to get the energy.
Right. And what is it going to do with this energy?
Yes, use it for the action potentials.
For instance, right? For the pumps, for the ion pumps, to make the action potential possible. The ion pumps burn ATP, adenosine triphosphate. The ATP is the most important energy source of our cells, and it is obtained by burning glucose, sugar, and oxygen. Cycle of Krebs, or Krebs cycle. All right. So let's say that I'm going to show you this visual patch in front of your eyes. Your visual cortex is going to do a lot of processing because this is kind of freaking out, making freak out your visual cortex. And what happens is that your visual cortex is going to tell your body, hey, look, I need a lot of oxygen right now because I have to work a lot, okay? And like I show you, this oxygen is gonna be used here with glucose to basically create energy, okay? All right, so yeah, so that's what happens. You have sensory, motor, and cognitive processing. You have the integration and signaling of a lot of neurons. They are burning ATP, right? To work, they need glucose and they need oxygen carried through the blood. Now, after this is done, you're going to have a displacement of deoxyhemoglobin, which is the residual of the process, right? And that's going to increase a coherent spin of the hydrogen nuclide diffusing H2O. Basically, it will create this magnetic disturbance in the signal, because we know that the signal is dependent on the water molecules, okay? But actually, how does it happen? Like this. Your brain is more active. It consumes more oxygen, and then you get higher concentration of deoxyhemoglobin with CO2. Carbon dioxide, that's how you call it, CO2, okay? The brain wants more oxygen. It says, look, I have to process a lot of things. I need more oxygen. And then the blood flow increases. You get more blood, okay? So the concentration of oxyhemoglobin, of blood with oxygen, raises. And that of the oxygenated blood decreases, okay? So this is a little bit the opposite of what you would expect. You would expect that the brain burns a lot of oxygen and then you have a lot of CO2 and then the signal, notice that? It's actually the other way around. When the brain works, you have an overflow of oxygenated blood because it is demanding more oxygen, okay? So at the end of the day, when the brain is working a lot, we get more oxygenated blood and less deoxygenated blood. And then if deoxygenated blood makes the signal fall, what is oxygenated blood gonna do? Make the signal? Anyone?
Rise.
Rise. So when the brain is more active, the fMRI signal rises. It will increase. All right, so this is basically it. And I'm really happy that we managed to do this. Right, so here you have, for instance, an envelope of how things work, right? When you have a baseline, you have some oxygen brought by your blood cells and some CO2. When you have activation, your brain is demanding a lot of oxygen, and then you get an overflow, an overburst of oxygenated blood. Okay, as you see here, you get way more oxygen than actually what you need in a normal situation. Okay, good. Now, the ball signal that I told you about is modeling this envelope of increase and decrease of oxygenated blood. And this is an example. You get your sensorial stimulation. Sorry. You have an initial dip, which is called by the initial consumption of oxygen, right? And then you have an overcompensation, which is all the oxygen then your body is sending to your brain. And then, right, so the concentration of the oxyhemoglobin increases, and then you have an undershoot, which is not due to the fact that your brain is burning a lot of oxygen, but the relaxation of the venous system. So your blood vessels relaxate in order not to be overpowered by the increase of blood. And then you have a temporary increase in the oxyhemoglobin. So this is called the hemodynamic response function. And then what the fMRI does is basically just taking this envelope and applying it to the predicted fMRI data based on the timing of your stimulus presentation. Okay? So basically it models the hemodynamic response functions to use to predict the fMRI signal. And yeah, so basically if these are all words, right? Word, 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 and a little bit of the pause and word, then you're gonna superimpose your bold envelope to all these particular pieces of stimulation. And at the end, you're gonna get something like this, which is basically the interpolation of this envelope repeated a lot of times. Okay? Any question at this point? Is this kind of clear? This is a little bit tricky, but the take-home message is that fMRI works because you get more oxygenated blood when part of your brain is active. All right, so let's see an example. Peterson et al., very important experiment, 1988. It used the subtractive method, which is a method, a methodology that is widely used even nowadays in fMRI. Okay, so here's the problem. How can we study a high order function which involves many subfunctions? An example, we want to see what happens when you recognize a word and when you retrieve the meaning of a word. Okay, so in order to recognize a word, you need to listen or to read. In order to retrieve the meaning, you need to be willing to produce a sentence or to have word recognition happen. In order to know what car means, you need to have grasped the word of car. You need to have turned the sounds that produce the word car or the graphemes into a lexical unit, which then is basically linked to some conceptual representation. Okay, good. So then how can we explore the pure meaning retrieval? Well, we just look what happens when you just recognize a word, and then what happens when you access the meaning of the word, and we subtract the process related to recognizing the word, or actually the fMRI signal, to the fMRI signal when you have to recognize and to retrieve the meaning. Now you see why it's called subtractive method, because you subtract what happens in a more basic process to a more complex process. Okay? So for instance, word recognition, passive viewing of words minus passing viewing of something that is not a word. Viewing words, because this assumes that viewing words is a composite process made by some more level process, visual processing, and some higher level process, word recognition, right? So this whole thing has a big flaw or a big problem, which is that a very strong assumption must be true. And this assumption is about the independency or the functional independency of these processes. Because if you start to subtracting things that happen when you think a process is going on to what happens when another process is going on, then if the two processes are modulated by each other, then you're going to mess up with the data, right? You see why, right? Let's say that the visual processing is going to give you a 1 point increase in the signal, and the viewing words is going to give you a 1.5 increase, but not because the word recognition gives you 0.5, but because when you sum 1 and 1, they actually interact, and the result is 1.5. And then you're going to get rid of 1, and you're just left on 0.5. Okay? So it's important that you assume that the processes that you're looking for have a relative level of independence. Okay, good. So here's what happened. In order to see what happened, where meaning retrieval happens, right? He had all these called experimental conditions. In one experimental condition, you just have a passive viewing of written words. The baseline is passive viewing of a fixation cross. And then, what about when you read aloud a written word? Well, then you must have had visual processing, word recognition, and also phonology articulation. So if you take the baseline, which is passing viewing of a written word, then you're going to subtract from here the same processes that are occurring when you have your baseline. So from your reading aloud, you get rid of the visual processing, you get rid of the word recognition, and you're only left with the phonological articulation process, which, guess what, is going to be localized in your motor areas, your areas that you actually use to articulate the message. Okay, good to know. So, whereas when we did the same for, yes, please.
Yeah, I have a question concerning the cross fixation. It implies that if we see a cross, we just don't give it any meaning to it, but it has a meaning, and it probably triggers some mathematical concepts and stuff. So it's not just a visual processing. There is also a recognition, another word recognition per se, but there is something happening, obviously, because it's not an anodyne symbol. It has a meaning, right?
Very, very, very true. So you just pointed out another problem, which is how you create your baseline, right? If you're going to show crosses, first of all, a cross is just one character, right? But the word is many characters. So a cross is simpler. Second, as you're saying, look, I know a little bit about mathematics. I see a cross, I think about sum, the operation of summing things, right? So that tells you that you have to be very, very careful when you choose your baseline or control condition. A better way would be to use meaningless symbols that have the same features of your word, for instance, the same length, the same amount of black, of ink used, right? But they are completely meaningless. And, you know, over the years, people actually refine this methodology, and we are going to see actually some cool examples about this. For instance, using Jabberwocky, which is a lot of words that have no meaning, that don't exist, but really looks like real word because they have the same morphological property. Okay, so yeah, very good point. Yeah, so let's forgive Patterson, who did this stuff in the 88, and let's give him some you know, let's be forgiveful. Any other question? Okay, so we are almost at the end, right? So we got to our saying words, right? So actually, when you did this trick with passing viewing of words, what would you activate is the occipital temporal junction, which is words minus cross. Someone yesterday asked me, where did you recognize written words? Well, this is a very important piece of the brain where you recognize written words. I mentioned like different gyri, probably I was wrong. This is probably the place, the occipital temporal junction, which is basically where the occipital lobe conjoins with the temporal lobe. Okay? Good, so let's move on. What do we get now? Well, we get to finally to the meaning retrieval. And what are you gonna do to explore meaning retrieval? Well, we have the baseline, reading aloud a written word. The only thing we need is to retrieve the meaning. So the way I did it was actually to generate an action. So if they saw the word cake, they had to say, what are you going to do with the cake? Eat. Okay? So according to them, this assumes that you access the meaning of cake in order to, figure out what you do with that. Now, again, this is kind of, you have to do a lot of stuff to go from cake to eat, a lot of planning, a lot of thinking, accessing some concepts and a lot of stuff. But again, let's forgive them. Sorry, let's, let's forgive them. And let's say that this is actually a nice baseline, right? And this is a nice task that pinpoints the retrieval of the meaning. Well, then what you do is just subtract your fMRI signal when you elicit this word production of this action-related word to the word you've seen, and subtract from this the baseline, which is reading aloud the written word. Therefore, you made an action of reading aloud the word associated with the other word. You get away the reading aloud part, and you're only left with the meaning retrieval, which, according to Peterson, activated this area here, which is something probably between BA47 and and some other area in the inferior frontal lobe. Okay, but you see that, by using this methodology, you actually have a nice guess about how to break down complex processes and how to investigate just a single component of a complex process, assuming that the independence of the process is all. Very good. To conclude, I'm just gonna show you another, so this is called subtractive method, okay? I'm gonna show you a different method, which is called factorial method. So here, instead of having 2 conditions or 3 conditions and subtracting one condition from the other, so subtracting a simpler condition out of a more complex condition, here you have 2 different factors crossed to each other. So for instance, you had these sentences like, the boy who's chasing the girl is Danny. The boy who the tall girl is chasing is Dan. Dan is the boy who the tall girl is chasing. Dan is the boy who is chasing the tall girl. What are these sentences? These are some object and subject relative clause, right? So the boy who is chasing the girl, who is referred to the subject, the boy, right? The boy who the tall girl is chasing, the boy, sorry, is the subject of the matrix clause, but it is the object of the embedded clause. It is the object of the relative, right? Who the tall girl is chasing the boy, right? Good. And these are typical center embedding relatives, right? Which has kind of a syntactic structure like this. Whereas the other sentences, they are right branching. So they're like, Danny is the boy who's chasing the girl, who's blah, 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 who blah, 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 who blah, 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 right? And they have this kind of down branching structure in the syntactic tree. You see the difference, right? We're going to talk more about this experiment, but just for now, just to show you that you have these 2 factors. One is subject versus object relatives, which here is called canonical word order and with the minus and plus movement, because when you have the subject of the matrix clause, which is then the object of the relative clause, then it has to be moved far away, and you see that in the red line, right? And then you have right branching and center embedding, 2 different kind of syntactic operations. Good. So in this case, what you're going to do is not to subtract your conditions, but what you're going to do is to investigate the effect of these 2 factors together. So if you just consider one factor, for instance, you can ignore these 2 conditions and just analyze your conditions for how they appear with respect to this factor, right? But then you can explore this factor in isolation, and then you can put these factors together. you see the difference between the subtractive design and the factorial design is that in the factorial design, one single condition, let's say this one here, right, is actually representative of the level of every factor of your design. These are also called nested factor or within items factor, okay? One single item is gonna bring the information of every factor in your design. This sentence here is center embedding and subject. Subject, pronoun, relative pronoun. This sentence here is center embedding, object relative pronoun. This sentence here is right branching, subject, right branching, object. Now, this methodology is more powerful than the other one because it allows you to generalize more deeply because, first of all, you're not throwing away data. And second, each single of your experimental items is going to bring information about all of your factors, okay? And the third interesting thing about this technique is that you can look at the interaction of these two factors. To see, for instance, let's say that object relatives are more difficult than subject relatives, center embedding is more difficult than right branching, then you might have two different predictions. One prediction is that these two factors are gonna linearly combine together, right? So when you get the most difficult condition in one factor and the most difficult condition in the other factor, you sum them together and you're gonna have a linear increase of cognitive struggle. But it might be that they actually do not behave this way. Maybe you are super fine if you get the more difficult condition of one of these two factors, your brain is going to be super fine, but then when you overload it with both difficult conditions, it's going to have an exponential increase in processing. Or maybe these two processes are processing two different areas of the brain, and then one area is going to be sensitive to one factor, and the other area is going to be sensitive to the other factor, okay? Do you see the difference between these two methodologies? Is this clear? Good. Finally, we have the parametric method where a factor, instead of being 1, 0, it's a parameter that varies continuously. For instance, in that design, the parameters were words per minute. No, sorry. I don't really know what this is. Right. So, yeah, whatever. We are going to see an example of this. Okay. Okay, good. So in this class, I brought inspiration from the Word chapters 3 and 4. Okay. And I have to credit these people who helped me with some of their slides. So if you want to know more about these things, please have a look at Word chapters 3 and 4. And if everything goes well, so please, sorry, I have to get rid of this insect. Think a little bit about the things that we did in these 3 days. Maybe go through the slides a little bit if you have time. So then the next time, if you have any question, you can ask me this question and we can discuss them together, okay? It's quite important that you understood not everything, not the details about what we did, but at least the basics. And today I was really happy that you showed understanding of the basics of the previous 2 classes. Then we should do the same with this one, okay? Because for instance, the next experiments we're gonna see, they're all fMRI experiments or something very similar to fMRI. So it's important to know exactly where we get this signal from, okay? Any question now? All right, then we are going to see each other on Thursday, right? Thursday we have we recover the class that we missed on the 1st of June. Okay, so no class tomorrow. See you on Thursday. I'm gonna stay here for a few seconds if you have some additional question. Thank you. Have a nice evening.
Goodbye.
Bye-bye.
Thank you. Bye.
Bye bye.